All right, hello everyone, and welcome to our next unit, Unit 4, and we're going to start with photosynthesis. All right, photosynthesis is one of those complex topics that we cover in biology over the course of the year, usually followed up with cellular respiration, which is pretty much exact opposite of photosynthesis. So everyone knows pretty much in photosynthesis we're going to take sunlight, carbon dioxide, and water, and turn it into sugar and oxygen, while cellular respiration literally is the complete opposite of that. So the two processes are really completely linked together. So what we're going to start with is several concepts that really are related to both topics that we're going to be discussing over the next several weeks. So let us get started. All right, so ultimately both photosynthesis and cell respiration are all about creating energy. All right, that is the key. So both these processes are going to share several concepts in common. And they're going to be what we call a hydrogen ion or proton concentration. ATP, so we're actually going to be making ATP in both photosynthesis and cellular respiration. And something called ATP synthase, which is an enzyme. Remember, anything that ends in an ASE we've discussed before is actually an enzyme. And then finally, something called redox reactions, which stands for reduction and oxidation. All right, with redox reactions, we're going to use something called electron carriers, and we're also going to make use of something referred to as the electron transport chain, or I'm always really going to refer to it as the ETC for short. So if you hear me say ETC, I'm referring to something called the electron transport chain. So again, both photosynthesis that we're going to start with and eventually cellular respiration are going to make use of these three concepts. Let's go through what we mean by these. So let's open up our energy toolbox here. All right, so let's pretend that those three things I just mentioned on the last slide are in our toolbox. So let's take one out at a time. First one we're going to start with is the hydrogen ion concentration. So when you see an H plus ion, again, all that really is at this point is a proton, because hydrogen atoms only have one proton and one electron. If they have lost an electron, they only have one proton left, so they are a proton. Now, what a hydrogen ion concentration is literally a collection of hydrogen ions that is created by the process of either photosynthesis or cell respiration and then stored within the membrane. All right, that stored hydrogen ion concentration is later then used to produce that main energy source that we've already discussed this year so far, and that is ATP, which stands for adenosine triphosphate. That's the fully charged battery. All right, ATP that all our cells want to use. So this is going to be found in both the chloroplast, which is where photosynthesis occurs, and the mitochondria, where cellular respiration occurs. So in both of these organelles here, we're going to create a hydrogen ion concentration, like you see here, that is then used in either photosynthesis or cell respiration. That's a process, a fancy term called oxidative phosphorylation, to eventually create more ATP. All right, so both processes need a hydrogen ion concentration. And really, to make it short and sweet, the idea is once you create a hydrogen ion concentration right here, it's like water behind a dam. This is the Hoover Dam. A large amount of potential energy is stored behind this dam. As you let some of that water flow through the dam, that water flow is being used to create ATP energy in our bodies, per se. So again, stored energy, which is the hydrogen ion concentration, is going to be a potential energy source needed to make ATP, which is what all cells want to use for their energy source. So again, the hydrogen ion concentration is like water behind a dam. Now, next, ATP and ATP synthase, the second thing in our toolbox. Again, ATP, as I've already said, is the energy currency of all cells. So here's an ATP molecule right up here in the top right-hand corner. We are going to make use of an enzyme called ATP synthase. Now, this same enzyme is in either chloroplast or mitochondria. They have the exact same enzyme in those two different organelles. And what ATP synthase does is, again, it's an enzyme, it's going to convert what we call rotational kinetic energy into the potential energy form of ATP. And as I've already said, it's found in both the chloroplast and the mitochondria. So as the chloroplast or mitochondria builds up this high hydrogen ion concentration. That hydrogen ion concentration is going to flow through this enzyme right down here. This is ATP synthase. So it's like water flowing through a dam. As it flows, and you can see the hydrogen ions coming down through ATP synthase right now, through the membrane, it causes this enzyme, ATP synthase, to spin like a turbine. And these are turbines you would find underneath the Hoover Dam. And instead of generating electricity or an electrical current, when this enzyme spins, that protomotive force is what they refer to it as, is providing the energy to add a third phosphate group back onto the uncharged battery of ADP, adenosine diphosphate, to make the fully charged battery called ATP, adenosine triphosphate. So if you think of it, let's see a little diagram here. Behind a dam here, we have our water potential energy, but let's pretend it's hydrogen ions. Those hydrogen ions are going to flow through the membrane, hit ATP synthase, which is going to start to spin like a turbine. All right, those hydrogen ions flow through the enzyme back to the other side of the membrane, which is now going to have a lower hydrogen ion concentration. However, that spinning motion of the turbine, or ATP synthase, in this particular situation, provides the energy again to add a third phosphate group back onto ADP to create our fully charged potential energy battery, ATP. And just like electricity then would go down power lines to the local cities, towns, etc., that ATP that we're now making can be used by any process within the cell itself. And the third thing in our toolbox is going to be what we call redox reactions, or the transferring of electrons. So there's two forms of redox reactions. There's what we call oxidation, in no particular order. Oxidation refers to the loss of electrons, and reduction equals the gain of electrons. So let's say you're really unhappy, and mom, dad, friend comes over, gives you a big hug, and now you feel better. Well, they just reduced your unhappiness or your sadness. So what's happening in your cells is multiple different molecules are always being oxidized, which means they lose electrons, or reduced, which means they're made happy again by gaining electrons. So here's a molecule here that has an electron that it's going to get rid of. It's going to give that electron over to another molecule. When it does, it becomes oxidized. The molecule here that accepts that electron, it's an electron acceptor, is now what we call a reduced acceptor. So it's made happy, and it now has the full complement of electrons that it wants to become stable. So both photosynthesis and cell respiration are really made, the whole processes are made to use both oxidized electron carriers and eventually create reduced electron carriers. How can you remember all that? It gets a little crazy. Think of an oil rig, all right, that you see out here in the sea drilling for oil. All right, oil could stand for oxidation is loss. So remember that oxidation always means that you're losing electrons, and rig, reduction, is gain. All right, so the reduction is the process of gaining electrons. All right, a lot of students always forget the difference between oxidation and reduction. Maybe oil rig will help you remember that on upcoming tests or quizzes. All right, some examples of redox reactions are, again, redox is oxidation and reduction together. Photosynthesis. So here's the general formula for photosynthesis that we're going to have to memorize at some point. Taking carbon dioxide and water. I would have light energy most times written here because that's the energy that is provided to convert these two molecules into glucose, as well as oxygen, which is a waste product. So what's happening in this process, carbon dioxide is being reduced to eventually become glucose, and water molecules are becoming oxidized to help create glucose, but also produce the oxidized waste product, which is oxygen. And then cellular respiration, again, is just the opposite. Flip that formula backwards. So now I'm going to oxidize or break down glucose, and I'm going to produce carbon dioxide, which is waste. That's what you're exhaling right now. So any food that you ate, literally a portion of it you were exhaling. And during the process of cellular respiration, we'll eventually see is oxygen is reduced. It gains electrons, binds to some hydrogen atoms, and becomes water. and water is a waste product of cellular respiration. All right, so each of these two reactions has both reduction and oxidation components, as well as each one is the opposite of one another. Electron transport change will be used. All right, so pretty much what these are energy staircases. All right, as electrons move down a staircase, that movement of electrons is used to generate a proton motive force, which is the hydrogen ion gradient. So look at it a little bit fancier here. So you're going to use that electron movement to create a hydrogen ion gradient, which can be used to make ATP. So this molecule here, NADH, carrying 2 electrons, can become oxidized. It loses its electrons. As those electrons move down several proteins, which analogy would be a staircase, that electron movement is used to create a hydrogen ion gradient that is used to make ATP. All right, and then you can use that ATP to do anything that you need in the cell. All right, even though this process of an electron transport chain, these would be integral or peripheral membrane proteins within either the chloroplast or integral or mitochondria, I'm sorry, electrons either obtained from sunlight or the breakdown of glucose are donated into that system, and as those electrons move down the staircase or chain, they help create a hydrogen ion gradient that is later used to make ATP. And that ATP can use to power the cell, or you can actually, in photosynthesis, the ATP is used to actually make glucose. And we're going to learn about that later in the week. When those electrons get to the end of this chain, they have to go somewhere. So they'll be donated to oxygen, which is going to now gain those electrons. So oxygen is going to become reduced and produce the waste product, water. All right, to finish up, about 2 more slides here. Again, we're going to make use of electron carriers, and I just introduced to you one of those on the last slide, NADH. All right, we're just going to remember what the abbreviated form of it is, NADH. Each NADH molecule can carry 2 electrons, or a pair of them. So NAD plus here, already jumping ahead, this would be an oxidized version. It's missing electrons. If it accepts 2 electrons, it becomes NADH, which is the reduced version. So as we're going to learn, first off in photosynthesis, we're going to use something called NADP+. This is going to be our oxidized electron carrier, and we'll use sunlight to create a reduced version of it called NADPH. And this will be used to make sugar later on. And in cell respiration, we're going to use 2 different types of electron carriers, but they'll both serve the same purpose. First, we'll use something that looks just almost like NADP+, but don't forget, cell respiration happens in the mitochondria, so not all molecules will be exactly the same, even though some will be used for the same purpose. So I have NAD+. It'll accept a pair of electrons, become NADH, which would be the reduced version you can see over here. And we'll also eventually, when we get to cell respiration, learn that we'll use a second electron carrier, an oxidized form called FAD or FAD. Once that accepts a pair of electrons, it becomes FAD2 negative. It'll bind to 2 hydrogens to become the fully reduced form of what we call FADH2. These 2 molecules here, NADH and FADH2, which are reduced, they'll be used to create a hydrogen ion gradient to make ATP. While in photosynthesis, NADPH, the reduced form, will be used to make glucose, which is a higher potential energy molecule. So think of NAD+, or NADP+, as an empty wheelbarrow. Once it gains a pair of electrons, it becomes reduced. The wheelbarrow is now filled. And to finish up this lecture, to kind of look at this whole process here, this actually what you're looking at is cellular respiration that will be occurring in a mitochondria. We're going to have to understand this step by step later on. But for now, just in conclusion, here is one reduced molecule of NADH, and here's another reduced molecule of FADH2. This will be along the inside of the mitochondria. Electrons from NADH and FADH2 create a hydrogen ion gradient as those electrons flow down a staircase here. And then that gradient will flow from high to low through ATP synthase that'll spin like a turbine to generate ATP.